Okay, so hi everyone. It is our absolute pleasure to welcome you all to this month's Kumustahan. For those of you that don't know, Kumustahan in Tagalog means to greet or say hello to someone. For AFSA, or the Association of Filipinx Scientists in America, we have regularly hosted Kumustahan to check in periodically with one another and feature another Filipinx scientist's work. Since my appointment as the Education Committee co-chair of AFSA, I've tried to put together a series of incredibly accomplished speakers to show us Filipinx and others in our communities that we can excel in STEM where we have traditionally been underrepresented. This month's Kumustahan represents the third month of our Filipinos in COVID series in collaboration with CFAM. In April, we had Dr. Lu Suriaga, who talked about early interventions in infants with HIV and the trends in HIV infection we see here in the US and in the Philippines. And last month, we featured Dr. Araneta from UCSD, who talked about the health disparities among Filipinx Americans, talking specifically about diabetes and COVID-19. We're absolutely excited to have you all here today for speaker, Dr. Valerie Arboleda from UCLA, who we will introduce in a bit. But talking a bit more about what AFSA is. So we are a network of Filipina, and Filipinx scientists in the US, which seek to utilize our collective experience and resources to support our communities in the Philippines and in the US. So I'll hand it off to my fellow peers from CFAM.
Hi everyone, I'm Shen. I'm an MD PhD student at NYU School of Medicine, and I represent the research committee of CFAM. We're a little over 2 years old, and basically we are the largest national organization for Filipinos in medicine. And we pride ourselves in connecting young Filipinos along the entire career path. So from pre-med all the way up to young attendings. In addition to these seminars, we hold a national conference every year. So welcome, and we're really excited to collaborate with AFSA. Jacob, next slide. Great, so we have a few rapid fire questions. And in the next slide, we just want to, if you can drop in the chat where you're located and what you're studying, medicine, or what your area of interests are if you're not specializing in anything yet. So maybe take a quick minute to drop your info in the chat. Nice. Nice. Well, it's great to see representation from all across the nation and even the Philippines. So welcome everyone. And our next rapid fire question, Jacob, just drop in your favorite Filipino dessert. Just choose one. I know you have a lot. Awesome. Well, it's so great to have everyone here and to get to know you a little bit. Next slide. Without any further ado, I'd like to just give you a little preview. It's my great honor to first, this is just our next Kumustahan. So if you want to quickly scan the QR, we'll also have this at the end of the seminar, but our next speaker will be Dr. Ponce on July 27th. So for today's speaker, it's my great honor to introduce someone I look up to as a physician scientist, a woman, a PI, a mother. She does it all. This is Dr. Valerie Arboleda. So she's an assistant professor in the departments of pathology, laboratory medicine, human genetics, and computational medicine at UCLA. She's what we call a Bruin born and Bruin bred. So she completed her MD PhD training at the David Geffen School of Medicine at UCLA, followed by her clinical pathology training at Ronald Reagan Medical Center at UCLA as well. She joined the faculty in pathology in 2018 as a physician scientist. And after receiving the NIH Early Independence Award, which was to leverage genomic technology to diagnose and identify treatments for very rare pediatric disorders, including Arboleda Lamb syndrome and Boring-Optis syndrome. She's an associate director at the UCLA Molecular Diagnostics Laboratories, and during the COVID-19 pandemic, led the FDA approval of SwabSeq, one of the very few scalable and sequencing-based tests for COVID-19 that are available. She continues to run her independent research lab and leads research and development for the UCLA COVID-19 SwabSeq testing laboratory at UCLA. Dr. Arboleda's incredible work has gained her multiple fellowship awards throughout her career, and most recently, her induction into the American Society of Clinical Investigation. So will you all join me in a virtual round of applause to welcome Dr. Arboleda?
Thank you so much, Sian, for the really nice introduction. I'm going to share my slides. Can you see my slides here? And you can see my shared screen?
Yes.
Okay, awesome. I can't see any of you, so if you're putting things in the chat, I don't know how I can see it, but I don't see them right now. So just feel free to unmute yourself and just ask questions, because I'd love it to be sort of interchange and dynamic. I'm thrilled to see this collection of groups around physicians and physician scientists and scientists coming together, because I have noted, and I've noted a lot as a woman, but also as a Filipina, that every time I meet a Filipino scientist or Filipina scientist, I get really excited, and I can count on my fingers sort of the number of professors I've met. It's probably like 2 in person since I started thinking about being a professor and was interviewing for faculty positions. So I'm really excited to see that pipeline grow and to see that you all are kind of looking into medical school. I have a number of classmates from medical school who are now faculty at various institutions. So we're around, but there are very few sort of, I would say, that are not in sort of exclusive clinical practice. There are very few of us. And so I think it's kind of just nice to kind of know you have other colleagues around who kind of understand what it's like to be Filipino and to grow up. And the vast majority of us are from California or live in California. So I know that I'm in a space where there are a lot of Filipinos, and I know I don't see very many. So I think that if for those of you who don't live in Southern California or Northern California, it's probably sort of even fewer and far between. So I'm going to talk to you today about some work that we did. I'm a pathologist by training, which means that I trained in exclusively clinical pathology, which is laboratory medicine. We're the physicians that you never see when you go to the doctor. When you go to the doctor, you get your blood drawn. Your tube of blood is labeled in a specific way and then goes into this black box, and you end up getting a result, I don't know, maybe a couple days later. And all of that work is done by a team of laboratory medicine scientists, so clinical lab scientists, but also pathologists who run and sort of ensure the quality of the lab and ultimately do a lot of the development of genetic testing along with our colleagues. So you can think of like clinical lab scientists as sort of the equivalent of my colleagues directly at the bench who do the day in, day out testing and make sure that it's of high quality. So that if you go here at UCLA and you get a COVID-19 test, and then you go to Utah and you go to ARUP and get a COVID-19 test, you can be sure that there's some consistency across clinical testing. So I'm kind of amazed you all want to still hear about COVID, because like, to be honest, it's a really tiring subject because it's been 2 or 3 years, and I can tell you the number of times I thought COVID-19 was going to be over, and we're kind of still in it. And today I was in the COVID-19 lab trying to like troubleshoot some new validations we're doing. So I think it's here for the foreseeable future. Our lab has a number of contracts for the California Department of Public Health for both COVID-19 and then thinking about sort of the future pandemics. And so I'm going to share with you a little bit about this sort of path we've taken over, oh gosh, it's been like 3 years now, 2.5 years to develop this test. So my research lab, so what you'll notice when you look at this slide is my research lab has nothing to do with COVID or really infectious disease. My training is exclusively genetics. I love genes, genomes, genetics, anything you can throw on a sequencer, I'm really happy. And I would say that like one of my favorite rotations as a resident in clinical pathology was around infectious disease. And the reason I liked it was because we sequence viruses, we do qPCRs, and that's sort of just where I feel very comfortable. And so I had done a lot of work in residency with our clinical microbiology director, Omai Garner, but I finished residency, I started my lab, and I kind of didn't look back. I wasn't really that interested in the diagnostic space. I did very little clinical work up until the start of the pandemic, where I was sort of in the middle of my research and we shut the entire lab down and kind of said, well, we can't really do anything because we're a research lab. The clinical lab is asking us for all of our reagents, but this is very strange that there's not enough testing, because this should be a very easy test to do. It's a virus, it has RNA, and we know how to amplify RNA. But what we came to realize is I learned a lot, I think a lot more over the course of the first 6 months of the pandemic about how test development works. And relative to sort of like what I learned in residency. And a lot of it was sort of trial by fire. So what you realize as we were sort of scrambling for testing in the first months of the pandemic is there's a lot of trade-offs in the world of testing. You can have a test that's really sensitive, so something that can detect down to 1 copy of virus per microliter. You can have something that's really rapid, so something like one of these benchtop PCRs, you do 1 test at a time, but it's not very scalable. So you'd need like thousands of these little machines that are quite expensive, and you'd have to run them all in order to do thousands of tests at the same time. And then you can have something like a sequencer. Like a sequencer can run billions of reads at one time. It takes a little bit longer, so it's maybe not as rapid, but it's very, very scalable. And so when we started thinking about testing, we kind of were like, well, this is weird. We do PCR in our laboratories and genetics all the time. Why is it so hard to find a clinical test? And so we can talk a little bit about that, and I'm happy to sort of touch on sort of my opinions there. But what you realize is that if you can find something that's scalable, rapid, and very, very sensitive, you've really found a unicorn. And I would highly suggest if that's something that you've stumbled upon, definitely go out and like patent it and get really rich, because that's kind of what it's going to bring you. There are no tests in my mind, not even the test that we developed. Ours is scalable, but it's not that rapid, not like an antigen test where your turnaround time is somewhere in the order of like 5 or 10 minutes, right? So when we started this, it was sort of like a couple of weeks into the pandemic, and a colleague of mine who had just closed his lab down at UCLA and moved up to the Bay Area and to start his technology, his startup, Octant Bio. And so they were using a lot of these high throughput techniques for drug discovery platforms. And so he reached out to some of his colleagues who happen to be chair of departments and have a bunch of resources, and said, I think we can solve this problem. I think we should be able to scale up testing if we wanted to, using sequencing based approaches. And it's essentially a qPCR, but with barcoded adapters. You can pool all of the PCRs, and this is what they use all the time for their drug discovery. And they were like, well, maybe we should see if we can get it working for SwapSeq. So people were trading qPCR machines because those were really hard to come by early on. And you can see this is a picture of his Twitter, just trying to like find a qPCR machine to do the early tests on. But that was sort of the ethos of the very early days. So, I actually wasn't part of that group when they started off. And then at some point, I think I happened to stumble across somebody, and they were like, hey, would you be interested in thinking about this with us? And I was like, sure, I'll join a meeting or two. And like 5 years later, we're still meeting like once or twice a week to talk about SwapSeq. And so most of you probably know that for COVID-19 testing, the gold standard right now is RT-PCR. So you reverse transcribe the RNA, you amplify it, and then you detect it by fluorescence. And generally, the limit of detection here is about 1,000 genome copies per mil of virus in swab media. But there's like a lot of logistical issues. And I think because we had never done the scale of testing as a society, the logistics were like, how do you accession that many samples? If you've ever, some of you may have worked in a clinical lab before or worked in a COVID-19 lab, people are manually accessioning samples. They're like typing in people's names. There's this RNA purification step, which was sort of the biggest bottleneck at the start of the pandemic, and it's really difficult to scale this up beyond a 96-well plate. It was hard to buy a QRT PCR machines. If you happen to be like at the Broad and they have like just a suite of QRT PCR machines that were used for other things, that then you sort of were in a good position. But if you were looking to buy them, the wait times were sort of out through the roof. Like you wouldn't be able to get them at the early times of the pandemic. And then supply chains. I mean, we still have these issues now, but at the start of the pandemic, you couldn't find swabs, you couldn't find tips for your machine. Even if you had the machines and the automation to do these things, you couldn't find the right tips to make them. And the price of the tests were expensive. So I would say that like at the time, and probably still now, insurance companies charge between 100 150 per test, but in reality the cost is far less, even if you're paying for all the regulations around a CLIA lab. And then there's of course turnaround time. So the assay time of the portion, the hardest part is actually getting all the samples into the lab at one time in order to run them at scale. So the technology that we developed is sort of has some features that are very similar to the classic technology of RT-PCR and fluorescence, but there are a couple key differences. So the first step is we obtain the specimen in a tube, either saliva or swab. The tube is key here, and we'll talk a little bit about it, but it's an automatable tube. So we can open and close it with a machine. We don't need to like open and close it, like twist it open and twist it shut. So even at UCLA, which is like a pretty high throughput lab, they still have techs who open and close these samples and pipette out a subset of it into another plate. We take this sample and we pipette it directly into the RT-PCR plate, and we then, which already has primers in it, the primers are all barcoded. And so as it amplifies the COVID-19 viral piece or your control, that barcode is attached and can identify which well that sample came from. And then what we do is, because we've now barcoded every single well, we then just pool everything together. So you go from these plates, you can pool up to 16 plates or even more at a time, and you're down to this 1.7 mil tube. You mix everything together, you purify it, and you can then run this straight on a sequencer. And the sequencing runs are about 5 to 6 hours because we only sequence a very short piece of the virus. And then it's all computational, which takes about 5 to 10 minutes before we can sort of deconvolute everything back to the original specimen and what the viral load is on that. And so there's a couple major differences. One is our readout is digital, it's sequencing. It's not based on a fluorophore that's turned on or off. You can use RNA extraction, but you don't have to. We do this thing called endpoint PCR. So we use all the primer that is put into that plate. So our primer dilution is actually really important. We do genomic barcoding of samples. So every single well has a unique dual barcodes on either side. And that's what allows us to run it on an Illumina sequencer and identify exactly which well it came from. And so these are all sort of important. That's sort of what makes SwabSeq really unique and specific. And so just so you get a sense of what we're trying to do, we have an amplicon here. You can add different barcodes to the end, and you can then identify explicitly where that sample came from. And this is sort of the unique part of our sample, because usually when you talk about pooling, most people think about pooling like you pool the samples before you do the PCR. So you take samples from me and maybe 5 people in this group, you put our spit together, and then you do the PCR to see if one of us has COVID. But our pooling is a little bit different because we have already labeled your sample before we pool, so we can tell exactly which one of the 5 of us has COVID. We don't have to say like, okay, all 5 of you now have to go get another test. And so that's sort of the big difference between sort of the pooling that people were talking about a lot at the beginning of the pandemic. The other thing that's really important, so any of you who have worked in genomic testing know that like when you're mixing samples together, when you're pooling different libraries together, the nanomoles that you pool together are really important, that if you have one library that's overrepresented versus underrepresented, then it can really throw off the dynamics of your actual assay. And so that normalization is usually what takes the longest when you're preparing a sequencing library. But what we ended up doing was we decided to add in a spike in. This is sort of like a positive control where we take what the viral sequence should be and we actually flip the first 6 base pairs. So what you can see here is in red, this is the actual viral sequence. And then here in blue is what we've sort of inverted so that even in negative samples, something gets amplified and we can actually digitally read it out. So we can actually detect if we're sequencing the spike in or the standard versus the actual virus and use that to normalize the amount of virus and sample that was inputted into the plate. And this is really important for 2 reasons. One, let me see if I can just. The first thing is it allows us to distinguish negatives from positives. So here, if you just look at the number of reads associated in a given sample, you can see that there's like, you can kind of tell the difference, but it's not super sensitive because the read counts can fluctuate a lot just based on other factors like inhibitory factors in the PCR. Everyone's spit has a little bit of like, maybe you ate like a potato the other day and it's now in our swab-seq reaction. But if you normalize this based on this to the spike or our standard, you can see that it tightens up a lot. You can see the positives really kind of stratify away from the negatives. And you have this really nice sort of linear graph where you can actually even call the number of viral reads that you have in that reaction. And so this is really important because it gives us a lot more confidence in our assay and it allows us to give a true negative, because if you see the spike, but you don't see the virus, you know your PCR primers were working. And this is sort of was one of the big sort of criticisms early on that we were able to address. So when we take this RNA purified sample, so a lot of people said, well, you need to make sure that you're looking at the same thing that our clinical lab is doing. So we did exactly that. We went to our UCLA clinical lab and we said, hey, can you give us some samples of patients that you've diagnosed with COVID? We want the purified RNA. Let's see how well swab-seq performs. And so you can see here in panel A, this is our contrived sample where we added some virus and we asked the question, at what level could we detect the virus outside, like beyond the negatives? And you can see here 250 copies per mil, so less than 1 copy per microliter. We can actually detect virus using swab-seq. Most of the QRT-PCR, and I put Stanford and Berkeley there because those are usually like our competitors, but for Stanford, for most of the qPCR sort of across the board, the best limit of detection you can get is about 1,000. As you look at different things like saliva direct and extraction-free methods, that number actually goes up to even like 20,000 copies per mil. So they're really just like, you lose sensitivity as you lose extraction, if you don't do an extracted method. And then panel B here shows a nasal swab of purified RNA. So this is a true clinical sample where we took a bunch of negatives, a number of positives, and we asked, like, can we detect the positives? And can we reliably detect a true negative? And the answer is yes. All of the samples that we tested that were positive and the negatives were negative. So we saw like pretty good concordance. Now, one of our main goals with developing this test, because it's not like we were getting paid, we weren't a clinical lab at the time, we wanted to just make it cheaper and faster. And how could we do that to not be limited by supply chain constraints? As we said, well, the biggest cost we have actually is the extraction protocol. It costs a lot on labor and it costs a lot to get the supplies, because at the time you were paying a premium for all of the machines. You had to buy multiple machines because no one vendor would give you all of their supply or as much supply as you needed. So we said, well, what if we just took the swab straight from whatever liquid we're dispensing it in and just ran that on a QRT PCR? And so we tried this a couple times. We tried taking the swabs into multiple different medias. And what you can see here is you lose a little bit on the sensitivity. So here we're down to like 500 copies per mil, whereas before I think we were closer to 250. And then we're still able to detect the positives versus the negative in real world samples. So you don't lose that much, right? Because 500 is still way better than the 18,000 or the limit of detection on antigen tests. And then, the other complaint that we were always getting was, oh, I hate sticking something up my nose. I don't want to do swabs. And so we were like, well, the other thing we can test is saliva. And for any of you who are have talked with the sort of pathologists and clinical microbiologists, saliva is like something everyone hates. They don't want to use it. They don't think that you can actually detect virus in it, and that's just an unreliable sample. And so we ended up saying, well, I know it's an unreliable sample here, like based on canonical thoughts and reasonings, but there's a lot of data around the world that says saliva is a perfectly good sample. We do oral swabs all the time. So we just did raw saliva, raw neat saliva, however you want to put it, into a funnel. The funnel, the saliva goes into the tube. It's kind of gross, but people seem to like it a little bit better, especially young adults, because you can just spit into a tube and then go instead of having to stick something up your nose and such. So what we did was, the one thing with saliva is that it doesn't amplify well unless you inactivate the RNases within saliva. So we actually have to heat up our sample to 95 degrees for 30 minutes. And if you're thinking about RNA, that's degrades our RNA into tiny little fragments. But the good news is SwabSeq is a really small fragment. It's the PCR fragment that we're amplifying is somewhere around 200 base pairs. And so that's fine. I don't care if we're fragmenting into smaller pieces. The problem is that when we move it towards, when we're trying to do like sequencing of the sample, it's just not as good. But what we ended up doing was we were able to show that saliva extraction free was able to give us a pretty good limit of detection. We're around 1,000 to 2,000 copies per mil. And then when we lose a little bit because we're comparing saliva to a purified RNA nasopharyngeal swab. So it's not truly like an apples to apples comparison. So what you can see here is that, like, you would expect that something where you're purifying the sample, you're also concentrating the virus there and you're removing all inhibitors. So SwabSeq is always going to perform better on the purified samples than on these sort of extraction-free protocols. But the piece that I think is key to remember here is that if you're looking to get really high throughput testing, you can probably give a little bit of sensitivity in order to make sure you're not affected by supply chains and having to batch samples delay samples by day, which I think a lot of people experienced over the course of the pandemic. So I would say, as a geneticist, the genomic technology works. It was a little bit complicated, but it actually is one of the more straightforward tests in my mind, because we can do a lot more with the genomic tech. But what I learned was that the logistics are sort of equally challenging. And I think if you had asked me 5 years ago if I'd ever be doing infectious disease testing, I would have been like, no, I don't think that's of interest to me. I don't really want to be involved in clinical lab stuff. But turns out just the training I had ended up being a real fit for this. And I was probably one of the few geneticists and pathologists who really thought that this could work. And so we went on this sort of bench to vending machine adventure, which I'll describe a little bit. There's a couple of things that I think are interesting and a lot of people don't really fully appreciate or understand, is that when you bring up a clinical lab, we're regulated by a number of, at the federal and state level. At the federal level, there's a piece of legislation called CLIA. It was enacted in 1988 with the intention of just ensuring quality across the country. So if you get a test here in California and then you go to New York, that, if you get a blood test, your blood test will be the same in both places within reason. And this is something that sort of the clinical labs take very seriously, because unlike a physician, so if you walk into my office and I see you as a patient and I'm writing a note, I see you, we're talking, and we have a whole conversation. But if you give your doctor a vial of blood and then that vial of blood gets to me, I have to do justice to that vial of blood, and I have no way of distinguishing your vial of blood from all the 30 or 50 other vials that exist. So tracking of samples is incredibly important, one of the big things in the CLIA labs, and then also making sure that you can ensure that your test is of high quality, that the people who are running your test aren't deviating from protocol. So it's a very highly regulated space. So when we developed this test, we actually were working out of a yeast lab. One of my colleagues who's a yeast geneticist who happened to have a MySeq. And so, we thought, well, that's fine. It's not like we're going to bring up a real test. And then eventually we were like, you know what, if we're actually going to do this, we can't be working out of this lab. It's a little bit too dirty. We need to be able to clean the space, and we need dedicated space. And so when we went to UCLA, we sort of asked them, do you have space? Like, do you want us to do this? Because if you actually want us to do testing for you, you're going to have to sort of like set it up, help us set it up. And so we ended up with dedicated space, which is just beautiful space. We had some funding from really generous donors. And then, honestly, at the end of the day, it was filling out a lot of forms. We ended up paying a company to help us develop our CLIA application and sort of like format our SOPs, our standard operating procedures for the lab so that we could obtain CLIA approval. And so CLIA comes through the state, but is regulated at both levels. And so our CLIA application went to the state of California, and they had to approve it in order for us to operate. And then the last thing is you need these certified clinical personnel. So not anyone can just walk in and say, okay, I'm going to run clinical testing. You have to, pre-pandemic, without the emergency orders, you have to go through a year of training. You have to have taken certain molecular biology classes in order to be a certified clinical lab scientist. Over the pandemic, they loosened those restrictions a little bit, but not completely. You still had to be compliant in like the number of people you had in the lab, the directors, and sort of their background and training. And so this is a kind of a nice picture. I think this is what everyone notices. We're on the 8th floor of the South Tower of the Clinical Health Sciences building. It was the old hospital from like 10 years ago, and they've remodeled it into lab space, but the 8th floor had not been used. And so what you can see here is like, these are our sequencers. They have this beautiful view of Los Angeles, and on a clear day, you can actually see all the way to the ocean. And when we started, we said, well, we don't want to have issues with supply. So we ordered a lot of equipment and a lot of supplies, and just said, who knows if we'll ever use this? Hopefully we'll have a chance to actually test people. But we didn't want to start testing and not be able to sort of provide supplies to all the people who needed them. And so this is the timeline. We started in 2020 of April, and we validated the technology within a couple months. We submitted our application to the FDA in like right before July 4th. So actually almost exactly 2 years ago now, 2 years. And at that point, we started thinking about how we might automate the whole process. So we started to hire lab staff. We started to pilot the design of our saliva collection kit. And once we had our EUA approval and our CLIA license, we were able to start deploying testing. And that was right before the Omicron surge in 2020, or the first winter surge. And it's just since then, we've just sort of been scaling up even more. We started off small, and we've been optimizing all along. And so one of the optimizations that you'll note here is that we really wanted to operate with like minimal lab tech, because we wanted to be able to run things as quickly as possible. And so what we had was we have all of our samples go into matching tubes. So it doesn't matter which tube you're using. So what you can see here is like, if you're doing a nasal swab, it goes into buffer. If you do saliva, you put your funnel into the tube and spit right into this tube that we register. This tube is barcoded, so it comes to us in a rack, and we can actually scan them all in together, and people will enter in their information on their cell phones, along with the barcode of their sample. And we go straight from here, we organize it a little bit, but then we inactivate the virus, so nothing is opened in the lab that is potentially like hot virus. We decap them, we pipette them, everything is like multi-channel to the max. So we're at 96 and 384 well pipetting. And the process has to be super simple or it doesn't work. So in the research lab, I love to tweak things, like to try and make it a little bit better, a little bit more efficient. But what we realized early on was that we have to make the process really simple and very straightforward. So for saliva, we were looking at collection kits that were cost like $7 or $8 a piece. Here we have a collection kit, which is literally a mini perfume funnel that I found on Amazon. And we ordered all of the mini perfume funnels you could find on Amazon in the beginning. And then we actually contracted with a factory in China to make us 1 million of these funnels. Because this is the easiest way. And like, we still got some complaints, but this made our testing really cheap, because our testing at the end of the day for a saliva test, we charge about $20 a piece. And then for our swab test, we designed a 3D swab because we couldn't get swabs. It was just impossible. So finally, we worked with one of the surgical residents at UCLA, who was in sort of our biotechnology, bioinnovation hub. And he helped us identify people to do a 3D printed swab where we had a breakpoint, and that breakpoint actually sits within the tube. So we don't have to remove the swab in order to pipette our samples out. And so those are a couple of the small innovations that we did early on that really helped us streamline our process. We've been through many iterations of quality improvement in the lab, but these are some of the key things. And then finally, I was telling you a little bit about how samples are registered. So what we have also tried to avoid is having any registration inside the lab. There's actually no HIPAA data that's like available to any personnel in the lab. So they don't see names, they don't see anything coming through. They just see tubes with barcodes. And so they scan them in, those get auto processed and sort of accessioned. And then they do their pipetting, and they're agnostic to whose tube is who. And I think that's a nice way to run the lab because we have no issues with HIPAA. We don't have any like patient information in the lab. It's all in a server that sort of gets matched with the sample at the very end. And even me as someone who's resulting, I don't ever see the patient information unless I actively look for it. And I haven't really had, there's no reason for me to look at who's testing positive for COVID. I result the result and that's it. Okay, so I'm close to the end. This is kind of what you saw where I left off the other one at 2020, but once 2021 hit, that's when we really started testing. We started testing for UCSB in November. We started testing for UCLA Health right when vaccinations were starting in the middle of December, and things were just insane. We didn't have enough personnel who were fully trained because we had just opened the lab, but we were testing thousands of samples a week and sort of at the same scale as our actual clinical lab that was doing inpatient testing for UCLA Health patients. So we actually ended up with a number of clients of people who couldn't find testing at that time. Caltech tested with us, UC Irvine, Pepperdine. We ended up doing some work with the California Department of Public Health. They were trying to sequence COVID genomes, and so when we had a little bit of a lull, we ended up doing more sequencing with them. And then finally, towards the end, in June of last year, I think we kind of said, okay, look how much testing we're doing. We're doing less than 1,000 like a week. We don't really need, we can probably like shut down the lab soon. But somehow the NIH we were chatting with the NIH and they said to us, they're like, we have forecasts that say that this is going to be around for a while. You may have more tests to do. And we were like, okay. And they were like, we'll give you a whole bunch of money to expand your facility. And we're like, well, we're not going to say no to money. Who does that? Like, we'll buy more sequencers. And if we use them for COVID, fine. And if we use them for other things, that's, that'll be great. We have access to sequencers. And so we had a NIH RADx award, which is a contract to expand the swab-seq facility. The goal was to be able to run up to 150,000 tests a day. And I think we can get to about 50,000 tests per day at this point, which is way more than we need, but it's something that we've had to sort of draw upon, especially when the undergrads come back to campus. And so this has been our testing volume over the last 2 years. We are over 1 million tests right now with swab-seq. I would say this only goes through March, but we're probably close to 1.5 million at this point. And it just keeps on growing. We've done almost 1 million tests for UCLA alone. I think it's at 900,000. So we've really hit a milestone for a lab that I would say that we weren't sure was going to be around for more than a couple months. So this is what you see when you walk around campus. There are a lot of these vending machines that host these COVID-19 test kits, and you just scan them in. You scan the QR code, you register your test, and then you spit into this tube and it gets taken to our lab. And so we've expanded to 50,000 tests per day. We have pretty significant automation. So we now employ, we used to just do mini-seqs and next-seqs. Now we have a NovaSeq, which hopefully we never have to use because that means it's really bad out there. But we do have a lot of automation as well. So just to save the pipetting for our technicians, and they've been amazing and tremendously grown over the past past year or two, just getting this automation up and running. So I can't, I think, take all the credit for any of this because I help with a lot of this stuff and I do a lot of the early work, but actually getting this operational is like a huge job. And I'd have to thank Don Marquette for doing a lot of this work along with the rest of the SwabSeq lab staff. And this is just a small subset of our team. And then we have a leadership group that really started off as a very small group pipetting in Leonid Krugliak's lab, who's our chair of human genetics. And so we started off, we were meeting 3 times a week for a good probably a good year and a half. I mean, we still meet once a week just to kind of keep things moving, but this has been the core group that sustained and developed SwabSeq and really kind of believed in the technology. And then if you want any more information, all of our preprints and all of our data and articles are all online, and I'm happy to sort of share any of those. I think Sian asked me to put together a little bit about my academic journey and sort of where I've been, and it's good to look back and kind of see the journey. I was born in Los Angeles when my parents came here from the Philippines. They immigrated in the early 80s. And then I was born, and then my mom at the time was studying for her board exams for residency. And so she is also a physician. She's now retired, but has practiced most of her life in Los Angeles. So I grew up in the Los Angeles suburbs. I did not want to stay in LA as an undergraduate. So I ended up in New York City, which I absolutely loved, but realized I'm not an East Coaster. I'm very much a California girl. This is just sort of, I'm much more laid back and happier in the sunny weather. But I do miss New York. And I stayed at UCLA for medical school, residency. And then in 2018, I started my faculty position at UCLA. So most of my time is spent in research. 80% of my time, I have 6 or 7 grad students at the moment, 7 grad students at the moment. I spend 10% of my time teaching, and then I spend 10% of my time doing clinical work. So that ends up being about 6 weeks a year. And my subspecialty is in molecular genetic pathology. So I actually, SwabSeq is actually on the side. I don't even count it because it's not through the Department of Pathology. I end up doing most of my clinical work within pathology in molecular genetic pathology, which I didn't do a full board certification in, but I did enough of my fellowship that they let me sign out these cases. And so my clinical load is fairly light, which is by design, because like you can't do everything at the same time. And then for those of you, I think this group knows well what an MD PhD is, but the traditional programs are these dual degree graduate programs. So you do 2 years of an MD, 4 to 5 years. I did 5 years of a PhD, and then 2 years of an MD. And these tend to be fully funded, so they pay you for tuition and living expenses for the entire program. They're long programs. They're kind of like marathons, in my opinion. So while they are fully funded, they are also just, you end up being much older than you realize you will be when you get out of these programs. And the real goal is to think about the next generation of physician scientists, right? To solve these hard problems. A lot of the challenges around the pandemic have been solved with physicians, scientists, and physician scientists starting to think about how we approach disease in the 21st century. So I'm just going to give a little plug for what I love about my job. I really like that clinical work informs my research. So for me, the flexibility after training was great. During training, it kind of sucked, my training was only 3 years, but the flexibility has been really helpful. I set my research agenda, I set my hours, when I work. It's stressful because if I don't do it right, I have a huge team of people who are sort of relying on me to bring in money and to set the direction. But I have a great team, so I can't complain. I really enjoy the teaching and working with students and postdocs. So that's kind of what brings me in. I come into the lab almost every day, and I actually really enjoy being at the bench. So the reason I enjoy telling the story of SwabSeq is because I actually did a lot of the bench work, because at the time you could only be on campus if you were doing COVID-19 research. And most of my staff, they were kind of uneasy about coming in. So I had myself and one of my, at the time she was a tech, now she's a graduate student at UCLA. And she came in with me, and we just side by side were pipetting and designing experiments. And it was so much fun. I kind of miss it, and I still want to do it. The things I don't love about my job, you can see here: paperwork, the FDA, and just like regular old bureaucracy of things that just don't work. So this is my lab. I think it has most everyone. It's not fully updated. But these are some pictures of the people in my lab pre and post pandemic. So it doesn't have the full set, and some of our funding sources. And then this building right here is the building that my lab is in. It's called the Biomedical Sciences, BSRB, Biomedical Sciences Research Building. So with that, that's really all I have for today. I'm happy to like answer questions and kind of see what you guys hear about what you guys are thinking.
Thank you so much, Dr. Arboleda, for that presentation and for all you've done during these past couple of years. It's really inspiring. We do know that you have to leave at a certain time point, but we want to ask you a couple questions if that's okay.
Yeah.
Angela, do you want to start us off?
Sure. Thanks so much, Dr. Arboleda. That was awesome. And I worked in clinical lab before going to medical school, so you made me kind of miss it a little bit.
A little bit.
Yeah, I love bench work too. So, okay, so we have a question. What is a unique challenge you faced as a woman of color, especially as a trainee, and how do you navigate those challenges?
So as a woman, and I think this is sort of universal to women, maybe and maybe compounded by a woman of color. I know that I look young, and so I think there's been a number of times, and I'm not sure this has really changed, but when I go to recruitment events to try and recruit, to like talk with students at the very beginning of the year when you're starting your PhD and people are looking for labs, I really get frustrated when people don't have name badges that have like who you are, because I always, I think, get recruited by a senior faculty member who's like, oh, are you here? Or I'll be talking with a group of students and they'll be like, oh, you're a group of students, no one's talking with you about their labs, let me help you. And I'm like, I'm chatting with everyone. And so that's sort of one that I always get. And I think one of the reasons I probably was less inclined to go into clinical medicine was because I would always get comments about how young I looked. And I never felt like the patients would really listen or kind of take me seriously. And so that was maybe one of the reasons that kind of pushed me more towards a research-based pathway. And it's a little bit, I find what always helps is when I'm on Zoom calls with more senior faculty, I will put my titles in my name here, as you can see, or when I'm concerned that I'm not going to get the recognition. So it's been helpful. So that's sort of one thing, is that I look young, I'm a woman, and I often get like asked to do things, sort of like the typical thing you would think of. And the other piece is that I find that a lot of the things I hear about happening have been really alleviated when when my department got a new chair. And so our new chair is a woman, she has kids, she's sort of been there and seen that. She's not a scientist, but I think she kind of realizes, she's like, well, I have a couple scientists in this department. I need to make sure they don't all leave, and they're pretty happy. So she doesn't really know how to manage scientists and like how to handle us. But at the same time, she kind of realized, like, as a woman, she's like, I just need to make sure that your name gets out there and that you hear about all these different events. And I think that's not necessarily mentorship, but sort of sponsorship. And she's and both my chair and the dean of the medical school, who recently left, Kelsey Martin. She left for New York to she's now the scientific director, I think, at Autism Speaks. She's at a big foundation. But she's been kind of one of those people who, when I was on meetings for SwabSeek, I was always the only one woman in the room, or one of two, right? And so I would often have people sort of talking over me, but even the chairs of departments who I know, some of them, like one of them at least, is married to another female scientist, I think. So he kind of realized, like, oh, Val, you had something to say, or you were trying to interject here. And you know, it's that kind of thing that I think has helped me a lot, because as a woman or as a person of color, you realize when you're the only person in the room that doesn't look like everyone else. But at the same time, I think I do thank my colleagues when they do things that I find really helpful. Like, I usually just thank them afterwards, because it's usually not something that they're cognitively, or like they're not necessarily aware of what they did that was super helpful, that I really appreciated, but I want to point it out so they do it again if they actually recognize it, right? Just to keep that sort of positive reinforcement going. And so those have been some of the real struggles is sometimes, and I pointed it out to people because we have it in our clinical lab. We used to have a manager because I stayed in the same institution. As a young woman, I was always I was the trainee, and then and so they would refer to me like Val this, Val that, do this, Val. And then as I became an attending and faculty member, it would still be Val. It would be Val and Dr. someone else. And I was like, but I'm also the doctor here. I'm the attending on record. And so sort of making that switch, some of it has been like, I just get annoyed by it. If it's like, I always make the conscious decision if it's worth my effort and time, because I can get annoyed and then just be like, okay, you know what, she's going to retire in 5 years and it's not even going to be an issue. Or it's something where I'll check in with some of my colleagues and be like, is this an issue for you too? Because a lot of institutions, you stay at the institution where you train. For a lot of us, and at UCLA in pathology, we see a lot of this, and they're all like, yeah, she does that to me too. She only refers to the older male attendings as a doctor, and everyone else is like Naroshi, Val, someone else. And I don't like to be upset all the time, so I do pick and choose which battles I think matter. And so I can say that the lab manager has retired, so I don't have to deal with that anymore. But whenever there's always sort of changeover, and you just get different issues. Is what I'm starting to realize, is that institutions will change and fluctuate over time, and you think, okay, this person has left, my life will be better, and you just sort of get something different. It's not better, it's not necessarily worse. And so I've just sort of chosen, that's why I spend a lot of my time in my research lab and in SwabSeq, and I don't spend a lot of time and energy and effort doing my clinical work. I do the clinical work that I'm supposed to do to maintain my skill set, and that's sort of it. And I kind of just sort of box that and say, like, I like some of my colleagues, the bench techs are okay, there's new leadership, and I'm not sure if they care whether or not I exist, and I'm okay with that. Like, I'm okay with actually sort of sliding under the radar, not having anyone ask me of things. Because once you start, I find this, once you start demanding things of people, they ask for things back. and so you sometimes want to make there are things where I think they're important and I want to be sort of at the forefront. And then the things where I'm like, I just need this for my skill set so I maintain my board certification. And I don't really want to do much more than the bare minimum. And it sounds horrible to say, but I think that's the reality of sort of, you have a lot of things on your plate. I have 3 kids. I don't know if I showed you their picture. They're very cute. And they take up a lot of my time. I don't really want to go home and be angry about all the various parts of things that annoy me at work. And some of them, I think the most I can do is sort of talk with people who are training with me and people who are working with me, and make sure that I set the right example, because eventually that will sort of propagate to the next generation. And I've sort of said, well, I'm an assistant professor now. Hopefully I will be tenured in a year or two. And sort of at that point, you can sort of make your voice a lot more known, and sort of institute real change at any institution. But you just have to remember that these are the things that bugged you when you were more junior and couldn't really say as much as you maybe wanted to. Because at the end of the day, it shouldn't matter. You should be able to speak up. But I've colleagues who are more senior vote on my tenure promotion within the department and within the university. And our department's super nice, and they have sort of said, oh, we always promote people. I haven't heard of anyone who's not promoted, but I have heard people say oh, that person's so annoying. They didn't like my research, or they didn't like this. and then, doesn't he realize that I'm going to vote on his promotion? And I'm like, ooh, okay. Note to self to kind of not rock the boat too much. Joshua, do you have a question?
Yeah, I was just curious on the science side, what your favorite discovery you've had when you were researching the lab was.
Oh, there's a lot of really interesting discoveries. So genetics is kind of just an amazing field. Some of my favorite discoveries actually herald back to grad school, which I think we're getting close to that in my research lab, but we were a little bit, I think, put back by the pandemic. So in grad school, I used to study disorders of sex development, which is a rare genetic disorder. And we ended up finding this syndrome called image syndrome. So it had been described by my PI And then we collaborated with somebody who had a large family of image syndrome. It has adrenocortical dysplasia. And we ended up doing one of the first like linkage analyses. And then we sequenced 20 base pairs of 5 family members and found one region that happened to have this mutation that was relatively rare. And I remember when I told my PI I wanted to do this, I said, I want to sequence this. It's 20 million base pairs. It's like 2007 or something. He's like, oh, that's too much. What are you going to do with all that information? You're never going to be able to analyze it. And I was like, well, I just want to try. So I like made a little I made a glass slide and we had oligos imprinted on it. And we sort of did it old school, technology wise, where we were putting it in an oven and rotating it. It was, I think I have the most fun with discoveries where I'm like at the bench and like you're the first person to like see the data and kind of go through that whole like laborious process. Yeah, actually talking with you guys makes me realize I do want to kind of go back to the bench. I've been keep saying it. I just have to clear out my schedule to do it. But that's one of my favorite discoveries.
Thank you, Dr. Arboleda. We know you have to leave at 5, but do you have time for one more question maybe?
I do have time for one more. It just takes me like 2 or 3 minutes to get to my car and stuff. So
Sure. Sharina, did you want to ask your question?
Yeah, thanks. Thank you for the wonderful talk. I was curious about, so I'm a core facility director at UNM. And so one of my colleagues is actually the one that's running the sequencing here. And so we're the flagship Trauma One Center for New Mexico. So he gets a lot of samples. I think like he would have loved to hear you talk. One of the problems that he said he was having was as more and more mutations are building and so many types of Omicron are happening, that even designing primers has been difficult because at least 30 different mutations just in the spike protein alone is making sequencing difficult and everything else. And I was wondering if you could speak to that, if you're sort of running into the same design problems as far as getting your primers to anneal. Thanks.
Yeah, so that's a great question. It's something that sort of keeps me up at night still, and that's actually why I was back in the lab trying to optimize, because we're revalidating a lot of our assay. So one of the downsides of the SWAB-seq assay is that we have primers to the spike, but we actually, most of the assays that exist right now have like 4 or 5 different primer sets. If we were to do that with SWAB-seq, we'd have to order like probably $1 million worth of primers with the barcodes in order to do what we do. Like our set alone, we can run, I think 6,000 samples, depending on how we combine the barcodes. And that set at the minimal scale we were looking at was something like $200,000 or $300,000 to get started. And so that's sort of been one of the limitations, is that we're always worried that we're going to have a mutation within our primer sets. So far we haven't. We've actually been really, really lucky that we haven't had a dropout for our samples. We know because we still test a ton of positives. Like we still get like 10 or 15% positivity rates when we're testing symptomatic populations. It is still that's one of the sort of the bigger problems with Omicron and with COVID, is that it just is constantly like evading as you get vaccinated and people are taking the monoclonal antibodies. It's just evolving. It's trying to get around all of these things we're throwing at it. So I don't know if there's ever going to be a solution, like a good solution to that, aside from having a lot of different primer targets. It's one of the development pieces in SWAB-seq is we're trying to decouple the barcodes from the primers so that you don't have to order each primer individually, like two per well, that you can then just order one set of primers and just distribute it across the wells and have unique barcodes that are going to be the expensive piece. It's a little bit of it's a challenge to do it that way. And it's a challenge to design the primers because you never know. So your best bet is to actually just like pepper them all over the genome. And even if one fails from a clinical diagnostic lab perspective, if one of your amplicons is positive, it doesn't matter if the rest of them were negative, you still call it positive. Even if your RNA, your internal control is negative, you still call it positive. For the sequencing.
I wanted to share something with you and everybody else that works in labs. A little bird told me that 2 of the largest China facilities that make filtered pipette tips, which obviously we need for all this type of technology, has shut down. And so USA Scientific is one of the leading manufacturers domestically, but they couldn't fill all the orders ever since those two Chinese manufacturers shut down because they were supplying all of South America. So USA Scientific was trying to float them, but now they said the demand's too high that they had to like cut all their contracts with South America. So when we talk about global supply chain, they were saying like, if your lab needs filter tips for doing whatever work they need, they're about to run out. So I just wanted to tell that I know there's a lot of people here that work in labs and need filter tips, especially for this type of technology. So just a heads up, if you don't have 6 months worth of supply, you may run out.
Oh, that's terrifying. I've heard that with those tips too, the thing that's actually really supply chain constrained is not the plastic for the tip, it's actually the piece, the filter piece. We keep that. This is why we keep about 6 months supply of everything in the COVID lab right now. So because we're running that's like 500,000 tests that we just keep on hand just in case. But my research lab probably should order some filter tips. So that is great. That's good to know. I'm gonna have to run and pick up my son from his daycare, but it was really nice to meet you all. And please feel free to like send me a message and if you're around LA, like if people are in Los Angeles and around UCLA, just let me know. I'm around here and I would love to I'd love to chat about science and such. And we have a lot of different projects. So if any of them are of interest, I love chatting about science and hearing what people are up to.
Thank you so much.
Dr. Arboleda.
Thank
Thank
you.
you all for coming.
Thank you.
All
All
right.
right, everyone. Oh, okay. All right, so can everyone hear me?
Yeah.
Yes. Okay, great. All right, so thank you for everyone that came to this amazing talk that Dr. Arboleda just did for us. If you're interested in getting to know more about AFSA or CFAM or joining, we have QR codes right here that will direct you to our websites, and you can also find us on Instagram, both AFSA and CFAM. And then we do have another Kumustahan coming up. That's going to be in July 27th, 7:30 p.m. Eastern, 4:30 p.m. Pacific, and the speaker will be Dr. Nina Ponce. And she is going to be, she works under the UCLA Center for Health Policy Research. And so the link for that as well is here to register. But thank you so much, everyone, for coming.
Are we all set, guys?
Yep.
Okay. Bye, guys.
Bye.
Hi, everyone.